Hello everyone, welcome to today's lecture on biochemical identification of bacteria. In this session, we will explore various biochemical tests used to identify bacteria and distinguish them based on their metabolic characteristics. Today, we will cover the importance of bacterial identification and its methods, the IMVIC test, including the indole test, methyl red test, Bouguere's-Pruskauer test, and citrate test, and the API kit, a rapid biochemical test system for bacterial identification. Bacterial identification determines the species and characteristics of microorganisms by analyzing their morphological and physiological traits. To achieve accurate classification, we use biochemical methods such as the IMVIC test and API kits. Molecular biological methods, including 16S rRNA sequencing, which provides highly precise bacterial identification. Bacterial identification is essential for differentiating microorganisms, verifying the authenticity or utility of bacterial strains, identifying disease-causing pathogens, which is critical for diagnosis and treatment. The IMVIC tests are a set of 4 biochemical tests used to distinguish members of the Enterobacteriaceae family, particularly between Escherichia and Enterobacter species. The acronym IMVIC stands for indole test, methyl red test, Bouguere's-Pruskauer test, and citrate test. Each test detects specific metabolic characteristics, helping us differentiate bacteria based on their enzymatic activities. The indole test is used to determine whether a bacterium can break down tryptophan into indole using the enzyme tryptophanase. This test is particularly useful for distinguishing E. coli from Enterobacter spp within the Enterobacteriaceae family. Tryptophan is an essential amino acid that can be oxidized by bacterial enzymatic activity. Some bacteria produce tryptophanase, which hydrolyzes tryptophan into indole, pyruvate, and ammonia. This test detects the presence of indole, which differentiates indole-positive bacteria from indole-negative bacteria. Kovacs reagent contains a reactive compound, which react with indole to form a red color compound, indicating a positive reaction. To perform the indole test, we first inoculate the bacterial sample into a tube containing SIM broths or tryptophan broths, both of which contain tryptophan. The inoculated medium is then incubated at 37 degrees Celsius for 24 hours, allowing the bacteria time to metabolize the tryptophan if they possess the enzyme tryptophanase. After incubation, we add about 0.5 milliliter of Kovacs reagent to the broths and mix gently. Kovacs reagent reacts with indole if it's present, producing a red color compound at the top of the medium. A positive result is indicated by the formation of red layer at the top of the medium, confirming that the bacteria produce tryptophanase and can break down tryptophan into indole. A classic example of an indole-positive organism is E. coli. If no color change occurs and the bird remains yellow or unchanged, it means that indole is not present, indicating a negative result. In this case, the bacterium does not produce tryptophanase, and an example of an indole-negative bacterium is Enterobacter spp. The methyl red test is used to determine whether bacteria perform mixed acid fermentation of glucose. It differentiates between E. coli and Enterobacter spp within the Enterobacteriaceae family. Some bacteria ferment glucose via the mixed acid pathway, producing stable acidic byproducts such as lactic acid, acetic acid, and formic acid. The pH indicator methyl red turns red in the presence of strong acid, indicating a positive result. If no stable acids are produced and the pH is above 6, the solution remains yellow, indicating a negative result. For the methyl red test, we start by inoculating the bacterial sample into MRVP broth, which contains glucose as a fermentable carbohydrate. The culture is incubated at 37 degrees Celsius for 24 hours, allowing the bacteria time to metabolize the glucose. After incubation, we add 5 to 6 drops of methyl red reagent to the broth. Methyl red is a pH indicator that helps us determine whether the bacteria have carried out mixed acid fermentation, producing stable acidic byproducts. The key here is that methyl red changes color depending on the pH. A positive result is indicated by a red color, which means that the bacteria have produced strong, stable acid. This confirms the presence of the mixed acid fermentation pathway, and an example of an MR-positive organism is E. coli. If the broth turn orange, this suggests the intermediate pH and is considered a weak positive result. A negative result is indicated by a yellow color, meaning the bacteria did not produce stable acid. An example of an MR-negative organism is Enterobacter spp. The Vegas-Proskauer test detects acetylene, an intermediate in the 2,3-butanediol fermentation pathway. Some bacteria ferment glucose using the butanediol pathway, producing acetylene as an intermediate. After incubation, alpha-naphthol and potassium hydroxide are added. If the bacteria utilize the 2,3-butanediol fermentation pathway, acetylene is oxidized into diacetyl, which then reacts with compounds in the medium, resulting in a brownish red color. If no acetylene is present, no color change occurs. In the Vegas-Proskauer test, we use the same MRVP broth that contains glucose as a carbon source. The bacteria are inoculated into the broth and incubated at 37 degrees Celsius for 24 hours to allow for fermentation. After incubation, we add 2 reagents to detect the presence of acetylene, an intermediate in the 2,3-butanediol fermentation pathway. We add 0.6 ml of 5% alpha-naphthol solution and 0.2 ml of 40% potassium hydroxide solution and mix thoroughly. The tube is left to stand for 15 to 30 minutes to allow the reaction to take place. A positive result is indicated by red color, meaning that the bacteria have carried out 2,3-butanediol fermentation and produced acetylene. This is characteristic of bacteria like Enterobacter spp. A negative result is indicated by yellow or copper-brown color, meaning that acetylene is not produced and the bacteria do not use the butanediol pathway. An example of a VP-negative bacterium is E. coli. The citrate test determines whether bacteria can utilize citrate as their sole carbon source. It helps differentiate citrate-positive Enterobacter spp from citrate-negative E. coli. Some bacteria produce citrate permease, allowing them to transport citrate into the cell. Once inside, Citrate is metabolized into oxaloacetate and then into pyruvate, releasing alkaline byproducts such as sodium carbonate. The pH indicator, BTB, changes from green to blue in alkaline conditions, indicating a positive result. For the citrate test, we use Siemens citrate agar, a defined medium that contains sodium citrate as the only carbon source and BTB as the pH indicator. We strike the bacterial sample onto the surface of the citrate agar slant and incubate it at 37 degrees Celsius for 24 hours. If the bacteria can utilize citrate, they will grow and metabolize it, producing alkaline byproducts such as sodium carbonate. This raises the pH, leading to a color change in the BTB indicator. A positive result is indicated by an agar turning blue, meaning the bacteria can utilize citrate as their sole carbon source. A typical citrate-positive bacterium is Enterobacter spp. A negative result is indicated by a agar remaining green, meaning the bacteria cannot metabolize citrate. An example of a citrate-negative bacteria is E. coli. API kit is a biochemical test system that identifies bacteria based on metabolic reactions. One widely used version is the API 28 kit, designed for gram-negative root-shaped bacteria. It differentiates 23 species within the Enterobacteriaceae family. The API test includes biochemical test, assimilation test, fermentation test. This rapid identification method is commonly used. In summary, today we explore the IMVIC test and API test, which are essential for differentiating bacterial species. Understanding bacterial identification is crucial for industrial application. Thank you for your attention.